Hello everyone, hope you all are doing well. So today we are going to discuss topic 30 of paper 2. Here we will discuss embryo rescue and detasseling. So first of all, we are going to discuss embryo rescue. What is embryo rescue? So it is the process when plant breeders rescue plant breeders rescue inherently weak, immature or hybrid embryo to prevent degeneration. So you can see from the name embryo rescue आप किसको रेस्क्यू करोगे? जो कमजोर है, जो कुछ खतरे में है. So here the embryos that are in danger, that are inherently weak, that are immature, that will be not be able to grow properly, we need to rescue those embryos. So that is what embryo rescue is. Now what are the advantages of embryo rescue? First of all, as we will do rescue, so it will help in the recovery of distant hybrids. Recovery of distant hybrids. It can also help in recovery of haploids. So when haploids will needed to be recovered when they will be in interspecific crosses, because during an interspecific crosses there are the chances that the embryo will not develop fully, so we need to rescue here. It helps in propagation of orchids. It helps in shortening breeding cycle, shortening or reducing breeding cycle. It helps in overcoming dormancy. Dormant embryo can be overcome. Here we can also do ovule and ovary culture. It helps to develop plants that are viable from weak embryo. Plants that are viable of weak embryo can be developed. Let me rub it. So it is very important in modern plant breeding. As we can see, its usage very important in modern plant breeding. It allows the development of interspecific and intergeneric food, or allows development of interspecific. I've just discussed interspecific, that is in the previous video I have discussed crosses between different species, and intergeneric, that is crosses between different genus of same family. So here we can develop interspecific and intergeneric plants or food. That is, we can also develop through these two methods ornamental plants. It helps immature embryo to survive. Immature embryo can survive here, as we will rescue this immature embryo. When this technique, that is embryo rescue, which is combined with molecular technology or molecular techniques, with combination of molecular techniques, it is used to incorporate specific traits, specific good traits such as resistance or of quality traits through gene transfer. It helps to overcome genetic variation in breeding line. Overcome genetic variation in breeding line. it helps in determining seed viability. It helps to study psychology of seed germination and development. Psychology of seed germination and development. So this was all about embryo rescue. Hope you all understand this. Now moving on to the next topic, that is, and this question has been asked in previous years, so it becomes important, that is techniques with special reference to intergeneric, interspecific and intergeneric hybridizations. So which technique are we talking about? Just I have taught, that is embryo rescue. So embryo rescue has been successfully used here in with reference to interspecific and intergeneric hybridization. So it has been used in, first of all, interspecific hybridization of genus Trifolium. It has been used in interspecific hybridization of genus Trifolium and Lycopersicon. It has also helped in intergeneric hybrids of Triticum, Aegilops, Sical, etc. You all have heard about these crops somewhere in breeding. It also helped in isolation of ovules. From interspecific crosses immediately after fertilization, and helped in regeneration of barley plants. And this experiment was done in Germany. So this regeneration was done in Germany, that is regeneration of barley plants. IRRI Philippines, that is Rice Research Institute of Philippines, I used this technique to transfer brown plant hopper resistance from wild species of Oryza, that is Oryza officinalis. So this is the wild species of rice, and from this we have got the brown plant hopper resistant gene, which has been developed and made it through crosses into normal rice, that is Oryza sativa. Now moving on, what is detasseling, the last topic of this session, that is detasseling. So what is detasseling? It is the removal of tassel. From the name you can suggest detasseling, removal of tassel. From female parent, that is mother, from female parent. It is done when tassel emerged out of boot leaf. When tassel emerges out of boot leaf, we need to detassel it. But before anthesis shed pollen, we need to detassel from boot leaf before anthesis. Because after anthesis the anthers will be formed and it will be difficult for us to detassel it. Anthers take 2 to 4 days to desize after complete emergence. but in some cases anther decides before complete emergence. So in that cases we need to detassel earlier, before this 2 to 4 days before the anthers decide, we need to detassel it. Originally detasseling is done after 14 days of emergence of tassel, up to 14 days of emergence of tassel. After this 14 days, and you can add 2-4 days for the time of decision of anther, but after anther has decised, it will be very difficult for us to detassel it, because anther has been decised and there will be chances of pollination. So there will be chances of mixtures or contamination. What is the significance of detasseling? To avoid normal pollination, first of all. After detasseling, we can use hybrid pollens. That is, here we see it can promote artificial pollination, hence can help us in hybridization. So through detasseling we are stopping normal or avoiding normal pollination. It helps in producing best corn seeds. So we know that farmers produce seeds for the next season. So if you know some practical knowledge, you have ever been in the fields, you would see that farmer used to detassel the crops so that the next seeds through hybridization are the best seeds. And after hybridization, after detasseling, the yields are increased by 5 to 6 times. 5 to 6 times the yield can be, and it is a easy procedure can be easily done in the farms. So this was the significance of detasseling. Hope you all understand this. So that is all for today. For any queries you can comment. If you like it, please press the like button, do share and subscribe. Keep making notes, keep value adding to your notes, as I tell you in every session that the notes you are going to make are your Brahmastra. More than 80 to 90% of the questions, almost every question will be directly from the topics I am teaching. So keep making notes. That's all for today. Have a nice day. Thank you.